Good afternoon, and welcome to the third of 4 U.S. Food and Drug Administration's industry webinars to discuss the infant formula transition plan for exercise of enforcement discretion. Today's webinar will provide more in-depth information on the quality factor requirements of sufficient biological quality of protein. I'm Lieutenant Commander Janisha Robb with the FDA's Center for Food Safety and Applied Nutrition, Communications, and Public Engagement staff. In a moment, I will turn the call over to Dr. Patricia Hansen, Deputy Director of CFSAN's Office of Nutrition and Food Labeling, to provide opening remarks. Then you will hear from Dr. Krista View, nutritionist in CFSAN's Office of Nutrition and Food Labeling, infant formula and medical food staff, to provide important information on FDA's requirements and recommendations regarding meeting the quality factor of sufficient biological quality of protein. For this webinar, we will not hold a question and answer session. Instead, we will bundle all questions related to the quality factor requirements and answer them during part 4 of the webinar series. Please send any questions that you have regarding the quality factor requirements and other details of the infant formula transition guidance through the registration link located in the YouTube description box below, and we'll be sure to address as many questions as possible in the final webinar of the series. With that, I will now turn the call over to Dr. Patricia Hansen, Deputy Director of CFSAN's Office of Nutrition and Food Labeling.
Thank you, Janisha, and welcome everyone to the third webinar in this series. As Janisha noted, in this webinar, Dr. Krista View, a nutritionist with our infant formula and medical food staff, will provide you with important information on FDA's requirements and recommendations regarding meeting the quality factor of sufficient biological quality of protein. As I've mentioned in the previous 2 webinars, the requirements for new infant formula submissions are the same, whether a manufacturer and its products are eligible to pursue the path outlined in our infant formula transition plan guidance, or a manufacturer is making a new infant formula submission through our regular established process. This includes the quality factor requirements. Most of our recommendations, the substance of them regarding meeting the quality factor requirements, will also be the same regardless of the pathway, although there are some process differences primarily around timing. In the last webinar, Dr. Carrie Asal provided an overview of the entire landscape of FDA's new infant formula submission requirements and recommendations, including the quality factor of sufficient biological quality of protein. In this webinar, Dr. View will be taking a deeper dive into FDA's requirements and recommendations regarding meeting this quality factor, with a focus on the protein efficiency ratio, or PER, rat bioassay. She will also touch upon the regulatory provisions that provide for exemptions from conducting a PER, provided certain conditions are met. We hope that all of you, regardless of the pathway you may be on and whatever your role might be in the process, will find this core information to be helpful. The information in this webinar may be especially useful to those involved in the design, actual conduct, or oversight of a PER study. And now to turn things over to Dr. View.
Thank you, Dr. Hansen. Good afternoon. My name is Dr. Krista View. I'm a nutritionist for the infant formula and medical food staff in the Office of Nutrition and Food Labeling at FDA. In this webinar, I will be providing information about the quality factor of sufficient biological quality of protein and the protein efficiency ratio rat bioassay, also known as the PER or PER. First, let's define a quality factor. The quality factors are those factors necessary to demonstrate the safety of the infant formula and the bioavailability of its nutrients, as prepared for market and when fed as sole source nutrition to ensure the healthy growth of infants. Demonstration of sufficient biological quality of protein is one of 2 required quality factors for a new infant formula. And protein quality can be defined as the ability of a protein to meet the essential amino acid needs of an animal, and that includes humans. 10696E requires that an infant formula meet the quality factor of sufficient biological quality of protein. It is important to note that this regulation states that the infant formula must meet this quality factor and not just the individual proteins. Protein quality depends on a number of factors and complex interactions. This includes differences in the digestibility of proteins from different sources, as well as the processing method of the infant formula. So thus, the quality factor was established for the protein within the infant formula matrix. The PER rat bioassay is the method by which sufficient biological quality of protein is demonstrated, unless an appropriate exemption request is made. The PER rat bioassay is described in the AOAC official method, and FDA has identified appropriate modifications to this method that are necessary when evaluating an infant formula. Please note that the PER study shall be evaluated prior to conducting a growth monitoring study in infants, as this permits a determination of the protein quality before the infants are exposed to the formula. 10696G includes 3 exemptions for this quality factor requirement that a manufacturer may request depending on the circumstance. It is the manufacturer's obligation to establish that all of the conditions of the requested exemption are satisfied, and exemption requests are reviewed by FDA on a case-by-case basis. Additional information on the quality factors, exemption requests, and infant formula submission requirements can be found in webinar number 2, titled New Infant Formula Submission Requirements and Recommendations. Next, we will discuss the AOAC official method protein efficiency ratio rat bioassay, which will herein be referred to as the AOAC method. The AOAC method was developed in the early 1960s to assess protein quality. For a new infant formula, the quality of the protein in the infant formula matrix can be compared to a protein of known high biological quality, such as casein, to demonstrate that the protein in the proposed formula is bioavailable and supports comparable growth in rats. The PER rat bioassay is currently the only method that accounts for protein digestibility and absorption in a living animal system. Now, the entirety of the AOAC method is listed on one page, and it covers the following topics: reagents or diet composition, experimental animals, assay groups, assay period, and calculation of results. It is important that PER studies are carried out under standardized conditions, most of which are described by the AOAC method. However, there are certain conditions that are not included in the AOAC method, such as the diet fed during the acclimation period, and we will try to highlight those that are and are not specified throughout this presentation. The AOAC method describes characteristics of the animal and the study groups. It specifies that the study include weaned rats between 21 and 28 days of age. The range of individual rat weights among animals used in the assay period should be less than or equal to 10 grams. Regarding the groups, for each test formula group, there should be a matched casein reference group. And please note throughout this presentation that the casein reference group will often simply be described as the reference group. Each group of rats must have greater than or equal to 10 animals, and many laboratories will include a 10 to 20% buffer in the event that something happens to one of the animals. The total number of rats should be the same in each group, and the average weight of rats in each group should not differ by more than 5 grams. The AOAC method does not specify a randomization procedure, but there are several methods available, and we often see it stratified by weight. Prior to the 28-day assay period, the AOAC method specifies a 3 to 6 day acclimation period. The AOAC method does not specify how the animals are to be housed during this acclimation period, though we recommend that they are housed individually in wire-bottomed cages during the acclimation period. We do this as it will facilitate the transfer to individual housing during the 28-day experimental period. Additionally, housing in wire-bottom cages throughout will reduce the rat behavior of consumption of feces, which is a behavior that may provide the rats with vitamins that are synthesized by intestinal bacteria. The AOAC specifies an assay period or experimental period of 28 days during which food and water should be available to the animals throughout. It is important that environmental conditions for both groups both be monitored and maintained as uniformly as possible to prevent potential confounding. Body weights of rats should be recorded on the first and last days of the assay period. Additionally, body weights and food intake should be measured at regular intervals, not to exceed every 7 days, and we generally recommend measuring at least twice per week. There are dietary components of infant formulas, such as lactose or proteins of lower quality, that can cause changes in the growth of rats during the study period. So we recommend that attrition and any adverse effects of the PER study diets are monitored. Ideally, this is done on a daily basis, but no less frequently than every 7 days. Some clinical observations you might find include oily coats, staining on hair, loose stools, and/or diarrhea. And all of the clinical observations should be recorded and included in the final report for the PER study. The AOAC method specifies that the following averages are calculated for each group of animals. This includes the 28-day weight gains, protein intake, and please note that protein intake is derived from nitrogen. The PER values, this is calculated as body weight gain divided by the amount of protein consumed during the assay period. And then the ratio of the test PER value to that of the Casein reference PER value. While the AOAC method is flexible in many respects, ensuring consistency in the diet composition between the reference and test groups is critical to ensure that the assay is not influenced by any uncontrolled differences between the two diets. The AOAC method describes the nutrient composition, which is listed in the table here, and formulas to ensure that the diets have a similar content of nitrogen, fat, ash, and crude fiber. The AOAC method specifies those 5 proximate analyses. A proximate analysis refers to the quantitative analysis of nitrogen for protein, fat, moisture, ash, and crude fiber. Carbohydrate is going to be calculated by the difference. So thus, we can consider these as the minimum specifications for chemical analyses in a PER study. Additional analyses can be performed if desired, and there are many methods available. As mentioned previously, a matched reference group is needed for every test formula group, and the performance of the concurrent reference group is equally important to that of the test group. So generally, data from historical reference groups are unable to replace the need for a concurrent reference group. One of the primary reasons for this relates to the importance of carefully matching the reference and test diets. Diets used in historical reference groups are unlikely to adequately match the composition of the infant formula-based test diet under evaluation. So next, we will detail FDA's recommendations for appropriate modifications to the AOAC method. Modifications to the AOAC method are necessary when evaluating the protein within an infant formula matrix. The modifications aim to ensure that the test and reference diets are as similar as possible in respect to the levels and types of fats and carbohydrates. Additionally, the vitamin and mineral content should also be matched. And please note, in this webinar, when we use the term match, we are not saying that they should be identical, but rather quantitatively similar enough, and here we are aiming for within plus or minus 20%. Without these appropriate modifications to the reference diet, the protein quality of the infant formula may be underestimated. Additionally, we must consider that the AOAC method was developed in the early 1960s, and there have been improvements in our understanding of rat nutrition since that time. So FDA considers that changes to the AOAC method that are based on such current understanding of the nutrient requirements of rats to fall under the appropriate modifications of the method. The protein concentration of both the test and reference diets is set to approximately 10%. Now, this is below the estimated level required for growth of 15%. By providing a lower protein amount, it ensures that the available protein is used efficiently, which improves the sensitivity of the bioassay. Now, as noted before, protein is calculated from the nitrogen content of the study diets. The AOAC method specifies the use of a nitrogen to protein conversion factor of 6.25 for setting protein levels. For the casein reference diet, there are several high-purity caseins commercially available for use, and FDA recommends that all specifications for casein be retained because it is helpful in confirming the amino acid content and adequacy of the diet. Now, due to the high fat content of infant formulas, the fat content of the test diet will be above the limit specified by the AOAC method. However, it is important to match the fat content of the casein reference diet to the test diet, even though it exceeds the limit specified by the AOAC method. Additionally, infant formulas contain a variety of fat sources, and FDA considers it possible to match both the fat content as well as fatty acid composition or categories of fats between the two diets. One reason to match the overall fat content as well as the categories of fatty acids relates to the vitamin E requirement. So the requirement for vitamin E increases with an increase in the consumption of polyunsaturated fatty acids, or PUFAs, as well as with the degree of unsaturation of dietary PUFAs. Therefore, we recommend that the total PUFA and vitamin E content of both diets be estimated and used to calculate the ratio of vitamin E to PUFA. At that point, appropriate modifications can be made as needed to prevent vitamin E deficiency. And a minimum ratio of vitamin E to PUFA of 0.48 has been suggested. Now, moving on to carbohydrates and specifically lactose. Now, lactose is common in milk-based infant formulas. However, we consider that products that are high in lactose represent a special category of foods, as rats do not tolerate lactose well and will often develop diarrhea. This can lead to an underestimate of the protein quality of the infant formula. So to limit possible confounding, the reference diet should match the lactose content of the test diet. Now, similarly, if the infant formula contains a carbohydrate source other than lactose, both the source and the amount of carbohydrate should be used in that reference diet as well. In addition to the composition of the reference diet, we must also consider how the casein reference diet is mixed or prepared. The preparation of the reference diet does pose several challenges because of the high fat content and sometimes high lactose content of infant formulas. So simply mixing the ingredients together may result in a reference diet that has a gummy or pasty consistency. This can be unacceptable to young animals, potentially lead to problems with poor digestibility and or reduced nutrient availability, ultimately contributing to reduced growth. So if it is possible, manufacturers might consider blending or combining the ingredients using those same general processes and equipment as are used in the manufacture of infant formulas. Once again, if that is available to you. And our thought is that the use of these procedures may help control for variables that are related to differences in the diet consistency between the reference and the test diets. Now, either liquid or powdered forms of an infant formula may be used in a PER study. If both forms of the formula are available, we do recommend using the one that is most processed, or the liquid infant formula. But we must keep in mind that infant formula is incorporated into the test diet based on its nitrogen content. And the AOAC method applies to products with greater than 1.8% nitrogen by weight. So due to the high water content of liquid formulas, they will not meet this minimum nitrogen requirement for the PER study. Therefore, they must be freeze-dried so that the test sample contains more than 1.8% nitrogen by weight before the test diet is formulated. Moisture is one of the 5 proximate analyses specified by the AOAC method. And it also provides a formula to adjust the water content of the diets to approximately 5% moisture. Now, please note the issue of water content of the PER study diets is distinct from the issue of drinking water that is provided to the animals during the study. As mentioned with other nutrients, the PER reference diet and test diet should contain a similar moisture content because the level of hydration may influence the PER study results. Additionally, the point at which water is added to the diets during the mixing process should also be considered, as this can also affect the PER values. We recommend that the issue of water in the PER study diets be discussed with diet formulators who have experience in blending diet ingredients to once again avoid any potential problems with diet consistency or palatability. The AOAC method provides the composition of a specific USP mineral mixture. However, it is flexible with the mineral mixture used in the PER study diets. So the following slides will detail considerations for selecting a mineral mixture, as well as FDA's recommendations for evaluating the mineral composition of the study diets. FDA considers the USP mineral mixture described in the AOAC method appropriate with the changes listed on this slide. The USP mineral mixture meets the estimated nutrient requirements for the growth of laboratory rats when used at 5% of the diet, except for the minerals zinc, selenium, and molybdenum. Additionally, the concentration of iodine, while not at a potentially toxic level, is about 200 times higher than its concentration in currently available mineral mixes. Lastly, cobalt is not required by the rat and can be omitted from this mineral mixture. There are other mineral mixtures available, such as the AAN76A and AAN93G mineral mixtures. However, we do not recommend use of one of the AAN mineral mixtures noted here without modification. This is because the 10% casein reference diet has a low sulfur content. Now, sulfur has not been classified as a required nutrient in experimental rat diets, but it is a critical part of the sulfur amino acids and vitamins. As you can see in the table below, the AAN mineral mixtures provide sulfur at approximately 0.3 grams per kilogram of diet. This is compared to the USP mineral mixture described by the AOAC method, which provides nearly 1 gram of sulfur per kilogram of diet. So the use of the AAN76A or AAN93G mineral mixtures without modification may lead to situations in which the inorganic sulfur contents of the final diet are lower than those described by the AOAC method. Therefore, we recommend that if using one of these AAN mineral mixtures, the diets be adjusted to provide the sulfate content set forth in the AOAC method. The AOAC method describes the analysis of ash as a surrogate for mineral content. Our current thinking is that ash content of the diet is not an appropriate surrogate for mineral composition when PER studies are conducted with infant formulas. Infant formulas contain significant quantities of minerals. And when matching ash content, the usual adjustment is to simply add less of the mineral mixture to the test formula than is added to the casein reference diet. Using this approach, the resultant ash contents of the finished diets is approximately the same. However, calculations of the individual minerals will likely show that the overall mineral composition of the test diet and the reference diets are not comparable. So rather, we recommend matching the mineral composition of the test and reference diets. And we suggest that you develop estimates of the compositions of projected test and reference diets prior to initiating the study so that you can ensure they are adequately matched. Regarding vitamin content, the AOAC method provides the composition of a vitamin mixture and specifies its use at 1% in both the test and reference diets. Therefore, the expectation of the AOAC method is that both diets will have the same vitamin composition, thus reducing the potential for confounding. However, the AOAC method does not provide a formula to describe how adjustments in vitamin composition can be made, and there is no single surrogate measure for all vitamins. So adjustments should be made on a vitamin by vitamin and formula by formula basis to match the vitamin composition of both diets. And once again, in this presentation, when we say matching, we are referring to plus or minus 20%. We do have specific recommendations for certain vitamins. The first is p-aminobenzoic acid. This can be omitted from the vitamin mixture. It is a vitamin precursor to folic acid and is not required by the rats. While also not required by the rats, vitamin C is one of 30 required nutrients in infant formula, and inositol is required in non-milk-based infant formulas. Therefore, we recommend that the vitamin mixtures contain these nutrients to match their concentrations in the test diet, even though they are not required by the rat. The estimated vitamin A requirement for rats has changed over the years, with the current requirement being approximately 10% of the vitamin A level that was specified in the AOAC vitamin mixture. However, infant formula typically has a high vitamin A content, and the amount of vitamin A in the reference diet should be matched to the test diet, even if it exceeds the vitamin A requirement of the rat. For vitamin K, menadione is specified as the source of vitamin K in the AOAC method vitamin mixture. However, it is approximately 1/10 as active as phylloquinone or vitamin K1. Therefore, we recommend that phylloquinone be used as the source of vitamin K in the PER study diets. And lastly, choline. Choline is included in the AOAC vitamin mixture, but it is not included in the AIN vitamin mixtures. As a required nutrient in non-milk-based infant formulas, choline should be included and matched to the concentrations present in the test diet. Thus, a separate addition of choline may be needed if using one of the AIN vitamin mixtures. The AOAC method described the PER study diets with 1% cellulose, which is measured as crude fiber in the proximate analysis. Now for the test diet, this ingredient is calculated as the 1% cellulose minus the fiber content of the test sample. The NRC does not provide a value for fiber in rat diets and only notes that the inclusion of fiber may be potentially beneficial. We know that infant formulas do not contain cellulose or other sources of crude fiber. However, they often contain non-digestible carbohydrates, for example, fructooligosaccharides or galactooligosaccharides. So our current thinking regarding the inclusion of cellulose in the reference and test diets is dependent upon the presence of non-digestible carbohydrates in the infant formula. So if the infant formula contains one or more sources of a non-digestible carbohydrate, our current thinking is that it is not necessary to add the 1% cellulose to either the test or the reference diets. However, when those non-digestible carbohydrates are present, they should be included in the matched reference diet and analyzed by appropriate specific methods. If the infant formula does not contain a source of non-digestible carbohydrates, we recommend that 1% cellulose be added to both the test and the reference diets. Going back to the topic of sulfur, methionine and cysteine, which are sulfur amino acids, are limiting amino acids in casein. Furthermore, cysteine, relative to methionine, is low in casein when compared to other proteins. So for example, the concentration of cysteine relative to methionine is only 14% in casein, where it is nearly 37% in skim milk. We know that when an essential amino acid is not available in sufficient quantities, growth and development are affected. However, despite the low cysteine content in the 10% casein reference diets, the AOAC method does not include the addition of cysteine. But as noted previously, the mineral mixture that is described in the AOAC method includes nearly 1 gram of sulfur per kilogram diet. Now, this may have contributed to sulfur metabolism and compensated for the low sulfur amino acid content. Therefore, we recommend that the PER reference diet contain the amount of sulfur that was specified in the AOAC method. And please note, we are happy to cover the topic of sulfur and sulfur amino acids in more details in meetings with firms that intend to conduct a PER study. Infant formulas often contain other components that are not normally found in rat diets. For example, ARA, DHA, nucleotides, etc. We recommend that the composition of the reference diet be as closely matched to the test diet as possible, and that includes these other components. And results of any analyses of these other components by appropriate and specific methods should be included in the final report of the PER study. So we've discussed the 5 proximate analyses that are described by the AOAC method, but additional analyses may also be appropriate. In fact, FDA recommends that manufacturers analyze several vitamins and minerals to ensure that the diet compositions are as anticipated. When selecting the vitamins and minerals to analyze, consideration should be given to micronutrients that may be present in different forms, or for example, if a separate addition was added, such as in the case of choline. Analysis of samples from different locations in the mixing bowl can also be helpful to assess the homogeneity of the diet preparation. An inclusion of records of the chemical analysis, both methods and results, can be helpful to verify the accuracy and appropriate of the reference diet and study diet compositions. Now, the AOAC method does not describe the diet that is to be fed during the 3 to 6 day acclimation period that precedes the 28-day assay. We recommend feeding diets of 10% casein, such as the casein reference diet, to both groups during this period. We found that feeding diets that have a high protein content, such as a standard rat chow, may introduce high levels of vitamins and minerals, which can make it more difficult for the rats to adjust to the PER study diets during the assay period. Rats should be weighed at the beginning and end of the acclimation period and the records maintained. As noted previously, lactose is not tolerated well by rats, and high levels of lactose result in detrimental effects on growth and PER values. So if the test infant formula contains lactose, it should be matched in the test and reference diets. However, to date, there is limited information on the inclusion of lactose in diets that are fed during the acclimation period. Earlier studies suggest that intestinal lactase activity declines rapidly upon weaning. Rats do not appear to adapt to high lactose diets. And lastly, they seem to tolerate diets that contain lactose at 20% or less. So considering the limited literature available, we recommend one of the 2 options listed here be used for acclimation diets when the infant formula contains lactose. So you could use an acclimation diet that contains 20% lactose. This would be used for both the test and the reference groups. The downside of this option is that it requires a 3rd diet preparation. And then option 2, you can use an acclimation diet that matches the lactose level in the test diet. So in this case, you would simply be using the casein reference diet for both groups during the acclimation period. And this option has the benefit of not requiring that 3rd diet preparation. We consider either of these options to be appropriate, and it's up to manufacturers and or the laboratory to determine which acclimation diet to provide. We do recommend the acclimation periods be kept as short as possible, remaining within the limits specified by the AOAC method. So review of the NRC nutrient requirement of laboratory animals at various steps during diet development can be helpful to assess the adequacy of the test and the reference diet. But please keep in mind that the purpose of the PER study is to compare protein quality with as few confounding variables as possible and to provide a short-term diet that either meets or exceeds all rat estimated nutrient requirements, as opposed to providing an optimized diet. So we recognize that there is limited information on the specifications for all nutrients in a 10% protein diet. The NRC nutrient values apply to 15% protein diets and are likely significantly lower for a 10% protein diet. So rather than using the NRC values to adjust the overall composition of the PER study diets, we recommend that they are used to identify and correct potential deficiencies and or toxic levels of any micronutrients. So next we will discuss results and record keeping. So earlier we noted that the AOAC method specifies that the following averages be calculated for each group. In addition, FDA also recommends that 28-day weight gain, food and protein intake, and the PER values be recorded and calculated for each rat in addition to each group. The AOAC method does not specify any statistical analyses to be conducted, though FDA encourages performing statistical analysis. We will commonly see descriptive statistics, such as the mean and standard deviation, used for continuous data, such as body weight and food intake, and analysis of variance is commonly used to compare the PER of the test group to that of the Casein reference group. Throughout the study, close study monitoring and record keeping are essential to understand the details of the conduct of the PER study and to aid in the interpretation of the results. This is particularly important in the areas of diet preparation, as well as for parameters that the AOAC method presents as ranges. We would like those values for the individual rats. And we recommend that complete records for all aspects of the PER study be maintained and that a detailed final report be prepared, which we will discuss next. The AOAC method does not mention development of a protocol or a final report, but we recommend that both be prepared. So the protocol is the planning document that can help ensure that the specifications of the AOAC method with appropriate modifications are met. And manufacturers with in-house facilities and contract laboratories with experience performing PER studies may have a protocol form that's available that can be used to develop such a draft protocol. We recommend that you submit your draft protocol to FDA for review prior to initiating the PER study. And additionally, for manufacturers with enforcement discretion that intend to conduct a PER study, the transition guidance recommends that you provide a draft protocol to review as part of phase 2 in the transition plan guidance. This will demonstrate meaningful progress towards establishing the quality factor of sufficient biological quality of protein. Additionally, FDA will review the manufacturer's draft protocol and will communicate our feedback. Some of the components of the PER protocol should include, 1, the infant formula specification. So the full specification for the lot or production run of the infant formula that is to be used in the PER study. This should include the quantitative formulation or recipe, as well as the nutrient content, expressed in units per 100 grams or per kilogram. And we do recommend that the minerals are expressed as their elemental forms as opposed to their salts. The complete specification of the infant formula is important because there are numerous calculations that are developed from it to confirm the diet compositions. Number 2, we also want the calculation of the diets. So once again, that quantitative formulation and the anticipated compositions for the PER casein reference, test diet, and acclimation diet if it differs from the reference diet. And we recommend including any specifications for casein, vitamin mineral mixtures, and other ingredients. Number 3, the diet preparation, so how the PER diets are to be prepared or mixed. And then number 4, the planned conduct of the live animal phase. So this includes the number of groups, selection criteria, and descriptions of animal husbandry practices. So regarding the final PER report, we recommend, once again, that complete records for all aspects of the PER study be maintained and that the final report be prepared. The report should include the following. You'll note here that everything listed on this page was also listed on the study protocol page. So the protocol and final report are closely related, and everything in the protocol should be reflected in that final report. In addition to these items, the final report should also contain the live animal observations, so looking for adverse effects or potential attrition, all individual live animal data, including body weights and food intakes, results of any chemical analyses, tabulation of results for both the groups and the individual animals, and a copy of the PER study protocol should also be attached. And we recommend that FDA review the final study report prior to initiation of the growth monitoring study. And the transition guidance recommends that a covered manufacturer that intends to conduct a PER study provide the draft study report to FDA for review as part of phase 3 of the transition plan guidance. In closing, appropriate modifications are needed to the AOAC method to accommodate evaluating the whole infant formula matrix. It is critical to match the casein reference diet and infant formula-based test diets as closely as possible, and here we are aiming for plus or minus 20%. We recommend monitoring live animal observations and evaluation of data as they are obtained, detailed record keeping, and preparation of study protocols and final reports. And lastly, we recommend that you request FDA feedback on your draft PER protocol prior to initiating a PER study, and the PER study report prior to initiating a growth monitoring study. And we do anticipate seeing both of those documents from covered manufacturers in phases 2 and 3 of the transition plan guidance. FDA will provide feedback in writing and or in a meeting. Thank you for your time.
Thank you, Dr. View. As a reminder, we will answer any questions related to the quality factor requirements in Part 4 of the webinar series during the question and answer session. Please continue to send any questions that you have regarding the quality factor requirements and other details of the infant formula transition guidance through the registration link located in the YouTube description box below. The final webinar in the series will be held on November 17th at 2 p.m. and it will focus on quality factor requirement, normal physical growth. This concludes today's webinar. A recording will be available on the FDA's meeting webpage shortly. Thank you and goodbye.